next speaker is a well-known and respected researcher in the CCHS community. The work from his laboratory and in collaboration with other Italian laboratories has elucidated the molecular action of FOX2B and the effects of FOX2B mutations on protein activity and aggregation in CCHS. We feel most fortunate to have a giant in the CCHS research field like Dr. Diego Fornasari, contributing his knowledge and talents to exposing the details of CCHS dysfunction. Dr. Fornasari is an associate professor of pharmacology and the head of the Laboratory of Molecular Pharmacology of the Autonomic Nervous System at the University of Milan. Dr. Fornasari graduated with an MD and PhD in pharmacology and toxicology at the University of Milan. He completed postdoctoral work in molecular biology at Yale University. His research focus is the action of the FOX2B protein in both the normal and pathological state. Dr. Fornasari is an active researcher with well over 30 papers published in peer-reviewed journals in just the last 5 years. Dr. Fornasari's work has been critical in modifying the general perception of CCHS as simply an incurable developmental disease. On the contrary, Dr. Fornasari, along with his colleagues, are convinced that an appropriate pharmacological intervention is possible and that it may help relieve symptoms and reduce the risk of death during sleep for CCHS patients. He most famously said at a previous CCHS conference, "It is not a matter of if CCHS will be cured, but only a matter of when." Dr. Fornasari is a staunch ally to the CCHS patient community. In this presentation, Dr. Fornasari will talk about the molecular pathogenesis associated with FOX2B mutations of CCHS. Thank you for your commitment and many contributions, Dr. Fornasari, and welcome.
Good morning to everybody. My name is Diego Fornasari, and today I'm going to discuss with you how to study the molecular pathogenesis of CCHS and what we have learned in many labs around the world about this very complex disease. First of all, CCHS, congenital central hypoventilation syndrome, is a disease of the autonomic nervous system. It is characterized by abnormal ventilatory response to hypoxia and hypercapnia, and this is the dominant clinical picture of the disease. But this is, as I said, a disease of the autonomic nervous system, and so many different dysregulation can affect this system. Temperature regulation, heart rate, disease of intestine can be present, such as a Hirschsprung disease, and in some rare, fortunately, cases, we could also have tumors derived from the neural crest. Basically, hypoxia and the response to hypoxia and the hypercapnia is are generally prevented during sleep, but in some patients it also depends on the severity of the mutations. Can have a problem with the control of respiration also during the day. This is a lifelong disease. At the moment, there are no pharmacological intervention, not just to cure the disease, but just to relieve some symptoms. And there is obviously the necessity of a ventilatory support that could be a nasal mask, a tracheostomy, or a pacemaker, the diaphragmatic pacemaker that can be used during night. The disease is related to mutation, so this is a genetic disease. So we have a mutation in a peculiar and particular gene, which is called FOX2B. FOX2B, as we will see very clearly in more details, is a transcription factor which binds to specific sequences in the DNA and regulates the expression of several genes, obviously expressed expressed in the autonomic nervous system and involved in different functions. What is exactly FOX2B? As I told you, is a transcription. It is a transcription factor, and a transcription factor generally regulates the expression of genes. Once the transcript has been made, the messenger RNA has been made, obviously we will have the production of the specific protein. So FOX2B regulates the expression of many different proteins in different anatomical districts belonging to the autonomic nervous system. Obviously, there are many different genes. We don't know very much about the target genes, the FOX2B target genes, but we know that some genes can be activated, so their expression, the protein that is encoded by that gene is increased. Sometimes we have a decrease in the transcription. So FOX2B is a modulator of the expression of many different genes. As I said before, there are no many target genes that we know, and this is an issue that we will discuss later, because obviously the mutations are in FOX2B, but many aspects of the disease are related to the altered regulation of the target genes. So maybe genes could be also target for a pharmacological intervention to overcome the effects of FOX2B. So this is the autonomic nervous system, and FOX2B is the master gene. So it regulates the entire circuits, which is made of different parts, including obviously the peripheral part, the autonomic nervous system, and among the different structure which has whose expression and formation are regulated in the autonomic nervous system, we have specifically these structures such as the retrotrapezoid nucleus or the nucleus of the solitary tract, which involved in the control of respiration. The disease is related to alteration in the formation or in the activity of this different structure. As I said before, respiratory control is the most important aspect of the disease, but this is a disease of the autonomic nervous system. So other aspects of the function of the autonomic nervous system might be involved. And for this reason, when we say to relieve some symptoms of the disease, we intend that we could maybe interfere with some symptoms, ameliorating the quality of life of our patient. FOX2B is not alone. FOX2B has a sort of cousin, which is FOX2A. FOX2A and FOX2B work together, and as I will show you later, they have a very important homology in the homeodomain. The homeodomain is this part here, which is responsible for the binding of FOX2B to DNA, recognizing this specific sequence. This is the core sequences. These are nucleotides which are present in the regulatory regions of the FOX2B target genes, and both FOX2A FOX2B recognize this ATTA sequence. So anytime, every time you see in the regulatory sequence of a gene this tetranucleotide, you can suspect that a homeodomain, a homeoprotein can bind there, and maybe just FOX2A or FOX2B could be involved. So FOX2A and FOX2B actually are partially overlapping in their control of the development of the autonomic nervous system, but some structures are regulated just by FOX2B, as you can see here in violet. In red, the structure which are regulated by both FOX2A and FOX2B, and here, but I'm not going to discuss this. There are two very peculiar structures which are involved with the ocular system, which are regulated just by FOX2A. So we said that the disease is related to mutations in FOX2B. What kind of mutations? Basically, we have two different types of mutation, PARMA and non-PARMA mutation. PARMA means polyalanine repeat mutations, because in the normal structure of the transcription factor, we have here a stretch of 20 alanine that in pathological condition can be expanded up to 13. So we eventually we will have 33 alanine residues here, and this is the reason of the malfunctioning of FOX2B, and these mutations are responsible for the disease. The other 95% of patients suffering from CCHS actually have this kind of mutation, expansion of alanine. 5% suffer from non-PARM mutation. That means missense, nonsense mutation, insertion, deletion, but not the expansion of adenine. Obviously, to understand something about the disease, some years ago, different labs started to generate mouse models in which FOX2B, the gene encoding FOX2B, was destroyed or was substituted with the most frequent PARMA mutation, which is the +7 alanine mutation. Different types of model have been generated, and what did we learn from these models? Basically, some important information that helped us to model the disease in humans. First of all, if you remove the FOX2B, you know that we have two alleles encoding a certain protein. If you remove both alleles encoding FOX2B, you have the animal die in a utero, and basically you don't have the formation of the autonomic nervous system. By contrast, if you generate a knock-in mouse in which the mutation, the mutant allele, the +7 allele, is inserted instead of the normal allele, so you have one allele which is normal and the other allele which is mutated, you have, of course, the animal die at birth, but you can start to distinguish which structures could be mainly involved in, because of this mutation. And these studies demonstrated that the retrotrapezoid nucleus is very important in the disease. By the way, since we are talking about respiratory controls, many other structures are involved. I mean, the retrotrapezoid nucleus is one of the structures that could be involved in respiratory control. So it is important to understand which is the condition, which are the conditions of this other structure, because maybe working on this structure could be possible to ameliorate the respiratory profile of these patients. So an additional complication to the story of FOX2B is that FOX2B regulates its own expression. That means that once the protein FOX2B is produced, this protein not only will regulate its target genes, but also will regulate the expression of FOX2B in itself. So just as I said you before, if you look at the regulatory region of FOX2B, you will see that there are many different ATTA tetranucleotides, and we tested the hypothesis that FOX2B could regulate its own expression, and our data, which has been published several years ago, confirmed this complication. And obviously, if you touch this ATTA site, you will lose the expression, or you will reduce the expression of FOX2B. So this ATTA ATA sites, tetranucleotides, which bind FOX2B are important for the expression of FOX2B. So basically the situation is like you can see here, we have 4 different ATTA sites in the regulatory region of FOX2B. Here we have the normal protein, which is produced by the normal allele, and the protein will go to regulate the activity, the expression of other genes, but at the same time it will regulate the expression of itself. But on the sick, on the mutated allele, the situation will be this. We will have an interference of the mutated allele on the mutated protein on the target genes, FOX2B target genes, but at the same time, we will have an interference of this mutated protein on the expression of the same of the wild type allele. So basically, probably the phenotype is not just the result of the interference of the mutated protein with the target genes, but also an interference of the mutated protein with the normal genes encoding the normal protein. So this situation for sure complicates the picture of the disease. To demonstrate actually that the mutated allele interfere with the expression of the normal allele, we performed some experiment. As you can see here, the expression of the normal FOX2B is hampered by the expression of the mutated protein, and according to the length of the mutated protein, you will have an increase in this interference. So basically, the plus 13 is probably the mutated form of FOX2B, which mainly interfere with the expression of the normal allele. Okay, given this picture, which hypotheses have been considered to understand the molecular pathogenesis of the disease? One observation was a very I would say the first observation, also considering this kind of mutation related to alanine expansion, was the fact that possibly the mutated version of FOX2B can form aggregates, and these aggregates can form in the cytoplasm or even into the nucleus, and these aggregates can entrap the normal version of FOX2B and can interfere with its activity in the nucleus. For this reason, the first part of the research in the field of CCHS were really focused on the possibility, on the demonstration of the presence of these aggregates, and at the same time, the treatment were focused to try to eliminate, to solubilize these aggregates, maybe sending in this way the protein in the nucleus. These are experiments from our lab, and basically these experiments demonstrated that the FOX2B, the mutated variants of FOX2B, can form aggregates in the cytoplasm and in the nucleus, but in vitro conditions, we thought that many artifactual conditions could be realized. So it depends on the amount of the mutated proteins you used, the time you wait before looking at the cells. So we were not, and we are not completely convinced that aggregation is the main mechanism by which mutated forms of FOX2B can cause the disease. This is part of the story for sure. We have aggregation and fibril formation, but for instance, also in vivo, we have never demonstrated the presence of these aggregates. Also in cadavers, it was not possible to demonstrate this. nevertheless, using in vitro context, we could demonstrate in collaboration with Emilia Pedone in Naples that actually the +7 mutation is able to form in vitro fibrils. So for this reason, we believe that this is one possible mechanism, but other mechanisms should be also considered to understand the molecular pathogenesis of the disease. By the way, the idea was to try to solubilize these aggregates, maybe sending the FOX2B into the nucleus, which is the place where FOX2B normally should work. And this is a paper from Isabella Ceccarini lab, in which, for instance, using these drugs, including ibuprofen or curcumin, it was possible to partially solubilize the aggregates and send the FOX2B, the mutated form of FOX2B, into the nucleus. By the way, we are not completely convinced that this strategy is a correct pharmacological strategy, because the formation of these aggregates maybe is a protective strategy that the cell use to protect, to avoid that mutated forms of FOX2B can just enter inside into the nucleus. And since these are mutated form of FOX2B, they can interfere with the normal function of the wild type gene. So there is a debate about this strategy, and we will see what will happen in the future. Other mechanisms could be considered to understand why this mutated version of FOX2B can interfere with the normal function of neurons localized in different areas of the CNS in the periphery. First of all, we could imagine that the mutated protein cannot bind correctly to its target gene, or we could imagine that the mutated protein does not correctly regulate the target genes. Maybe there are also aberrant target genes that are recruited by this mutated form. We have the problem of the dimerization. We know from our lab and other labs that generally FOX2B does not work alone, but it works as a dimer, as an homodimer. This was what we knew until a few years ago, but probably also other proteins such as FOX2A could be involved in the formation of heterodimers. And finally, as a transcription factor, FOX2B has to interact with other proteins, and maybe the interaction is aberrant. So concerning the binding to DNA, we have demonstrated that for sure, especially with the +13 alanine expansion, we have an interference with the binding to DNA. But basically, we can say that there is a slight decrease in the binding of the different mutants to DNA, but this is for sure not the most relevant mechanism by which FOX2B can interfere, with the exception of +13. So basically, we have a decrease, but this decrease does not abolish completely the binding of FOX2B. And here, these are biochemical studies in which you just by gel shift assay you can see the binding of FOX2B to its ATTA binding sites. These are functional studies in which specific target genes have been tested with mutated forms of FOX2B to see if there is an interference with their transactivation, with their activity. And as you can see, it depends on the type of target gene you consider. For instance, with the DBH promoter promoter, you have a severe decrease of the expression of the DBH in presence of the different types of PARM mutation. This situation is less drastic with the FOX2A promoter, but what is interesting is that if you add the normal form of FOX2B, you rescue in some cases completely the normal activity of the target gene. So it means that if we could increase the expression of the FOX2B normal allele, probably we could block the activity of the mutated form of the protein, and we could restore some functions. Another aspect that we have considered is the formation of dimers. As I said before, FOX2B does not work alone. FOX2B works as the dimer, and we believed for years that the dimer was formed by two molecules of FOX2B, so two in a homodimer. But our experiments and some experiments also in other lab suggested that actually FOX2A is the privileged partner of FOX2B. We don't know in vivo, but for sure in vitro. I'm going to show you some very impressive results about the formation of heterodimers between FOX2A and FOX2B. This is the molecular strategy. I don't want to enter into details. Obviously, the strategy we used to study interactions between FOX2B and FOX2B and FOX2B and FOX2A and FOX2A and FOX2A, and we published these results some years ago. And basically, what you can see here is that for sure we have the formation of homodimers between FOX2B and FOX2B. But if you look at this and you look for the most efficient dimer which is able to regulate transcription of target genes, you will see that the most potent combination is between FOX2A and FOX2B. This is the combination, the homodimer, FOX2B and FOX2B. But what is interesting and relevant in this, we don't know if this is the privileged combination inside the nucleus, inside cells. But we observed this interesting situation. As you can see here, if you have maybe you could observe this aspect also here. If you have a combination between the FOX2B normal normal protein and FOX2B mutated protein, in this case +7, you have a decrease of the activity of the dimer. So when the dimer is formed by a mutated form of FOX2B and the normal FOX2B protein, you have a decrease, you have a drastic decrease in the activity of this combination. By contrast, if you use combination between FOX2A and, for instance, you can see this very well here. This is the combination with the wild type FOX2B. If you have a dimer between normal FOX2B and +7, doesn't work anymore. But if you have FOX2A and +7, you rescue the transcriptional activity. So I'm saying that as a FOX2B, also FOX2A can rescue the activity of the mutated form of FOX2B. And so this becomes an interesting target, because if we could increase the expression of FOX2A, we could get some rescue and some relief of the transcriptional defects which are associated with mutation in FOX2B. This is quite, you see here, FOX2A +7, and this is FOX2B +7, okay? So this is other possibility we have to try to correct, to rescue some of the function. Another issue which is very relevant is related to the target genes. As I told you before, the disease is for sure related to mutation inside the gene encoding FOX2B, but basically this mutation will alter the expression of the FOX2B target genes, and eventually the disease will be related to alteration in the expression of the FOX2B target genes. So you could imagine that you could also bypass FOX2B and try to correct the defects on its target genes. As I told you before, there are very few genes which are known to be target genes of FOX2A and FOX2B, and for this reason, a few years ago, we decided to use a genome-wide approach to identify the possible target genes of FOX2B. The strategy is called chromatin immunoprecipitation and allowed us to identify univocally genes which bind FOX2B, and with other experiments we could demonstrate that some of these genes actually were also transcriptionally regulated by FOX2B. FOX2B is a very important regulator of many genes which are normally expressed in the circuit of the autonomic nervous system, and obviously we could not study all these genes, so we focused on some of them, and we started with potassium channels. FOX2B is an important regulator of the expression of potassium channels, and potassium channels are very relevant not only in the normal physiology, but because mutations in potassium channels have been frequently involved in genetic disease affecting respiration. So it was an interesting field to explore. Potassium channels basically are this. Generally, potassium is more concentrated inside the cell than outside. And when you open one channel, one potassium channel, generally potassium will leave the cells. But potassium is a positive ion, and in this way, the cell, the transmembrane potential of the cell will become more and more negative. And when the cell is hyperpolarized, it means that it is not very reactive from an electrical point of view. If you have many open potassium channels, probably the cell is dormant, is sleeping, because it is too much hyperpolarized. Keep in mind this. We demonstrated that actually FOX2B is able to regulate the expression of many different potassium channels. We also investigated sodium channels, and we are doing the same with calcium channels. So if you remove, it's interesting, look at this. If you remove from cells FOX2B, some potassium channels are increased. So it means that if you have a defect in the activity of FOX2B, maybe in the cell you have an increased expression of potassium channels, and these cells become dormant, become less reactive to external stimuli. Let me speculate. Let me imagine. Maybe they do not respond properly anymore to carbon dioxide or oxygen and so on. Obviously, this is a molecular demonstration, but we didn't know anything about the real role of these channels in cells. So for this reason, we ask an electrophysiologist, Flavio Antonucci, in our department, to tell us if the manipulation, the genetic manipulation of FOX2B could actually affect the electrical properties of the cell. And what Flavia demonstrated is that when you remove FOX2B and then you reintroduced the wild type variant of FOX2B, you have a variation of the passive and active properties of the plasma membrane of neurons, which are in agreement with the fact that you are modulating potassium channels. So probably there is a real link between FOX2B and the regulation of channels in a way that the cells maybe become less responsive. You know, this way to think the experimental approach. We say that there is one road from bench to bedside. That is exactly what normally scientists do, but there is also a different and opposite road from bedside to bench, and generally observing and considering the clinical aspect of the disease for us it's very important, because we could test hypotheses at the bench deriving from observation in the real life of the disease. I'm basically talking about this serendipity, the finding that many years ago, the finding that 2 CCHS patients using desogestrel, which is a progestin drug, had an increase, an improvement, sorry, of respiratory performance. I heard that 2 different studies have been started in the past, which has been interrupted. I don't know why. And this hypothesis of the role of desogestrel, of the possible role of desogestrel in CCHS has been left and abandoned, and for many years we didn't discuss any more about this. But our hypothesis is from the bedside to the bench. So we started to investigate if desogestrel had any relationship with the activity of FOX2B. You know that desogestrel works on the progesterone receptor, which is a transcription factor. So it is a regulator of gene expression, exactly as FOX2B. FOX2B is a transcription factor regulating the expression of many different genes. And some years ago, we could demonstrate that a precise link exists between FOX2B and desogestrel, because desogestrel is able to downregulate the expression of FOX2B. We described in details the mechanism by which this occur, the importance of the presence of the progesterone receptor, and so for sure there are some effects of desogestrel on the biology of FOX2B, and not only we have a decrease of the expression of FOX2B, but we have also a decrease in the expression on its target genes. So it's a cascade. Desogestrel, by regulating FOX2B, is able to regulate also the expression of the FOX2B target genes. But more important, desogestrel is able to downregulate also the expression of the mutated form of FOX2B. So this is an interesting observation, because maybe a balance between, by correcting the balance between mutated and wild type proteins, might restore some functions inside the cells. Obviously, these are in vitro experiments, so we ask to Silvia Pagliardini and with the activity of the activity of Silvia Cardani, who left our lab to join Silvia Pagliardini in Alberta, in Canada. They demonstrated that in animals, so in vivo, you can regulate by etonogestrel, which is desogestrel substantially, the expression of FOX2B and its target genes in some nuclei involved in respiration, in this specific paper, which has been recently published in the solitary tract nucleus, which is a relevant nucleus for the control of respiration. So for sure, desogestrel is involved in the biology and in the regulation of FOX2B is able to downregulate the expression of the mutated form of FOX2B. And for sure, this is an interesting approach just to study the disease. We don't know yet if we will never use the desogestrel or derivatives of desogestrel in the treatment of the disease. When I say treatment, I intend to relieve some symptoms, to put patients in an area of safety increased, for instance, concerning the sleep and so on. We don't know that, but we know that the desogestrel in our hands is very, very useful to understand some aspects of the disease. Another, obviously, the question is, but is desogestrel able to regulate the expression of channels, as I demonstrated before? The answer is that we have several channels which are upregulated and several channels, including potassium channels, which are downregulated when you expose cells to desogestrel. So we have a modulatory effect of desogestrel on the expression of channel and on the electrical property of the cells. And finally, with Flavio Antonucci, we just could demonstrate that in itself, desogestrel is able to increase cell excitability. That means that a dormant cell can become or can come back to an active state. So many aspects of the puzzle go together. But obviously, we always try to be closer to the disease. And so beside in vivo models, we started to get to modeling the disease by using stem cells obtained from patients. We are not alone in this. The lab of Gadvatin obtained stem cells from patients, and we are doing the same, and probably the combination of the different information arising from these two labs will help to better understand one more time the disease. I'm going to describe the data obtained in my lab, and basically we got by biopsy, and I have to thank families and patients because they help us with this very, very much, and they believed in this project, and we are in debt with them. And by biopsy, we got fibroblast, and we reprogrammed this fibroblast. We went back to the state of stem cell by using some tricks, and finally we got the neurons. Obviously, you have to decide what kind of neurons you want to study, and we decided to study sympathetic neurons for the reason that I'll tell you in a few slides. We have published the paper very recently, and basically we got stem cells from two patients bearing +5 mutation, but what is interesting is that one patient is an early and the other one is a late onset disease. So same mutation to understand the clinical pictures, which is different in these two different patients. Maybe not all the audience is familiar with stem cells. Basically, stem cells are our origin, our organism develop starting from cells which are completely undifferentiated and can become whatever they want. They can become neurons, muscle, skin, and basically stem cells are inside, are the starting point in the human fetus. You see here cells, and these cells will become specific organs. In the past, it was believed that it was not possible to go back from cells which have been already differentiated back to stem cells. Actually, we know that this is possible. This is the strategy that many labs now are using, basically taking somatic cells and with a mixture of appropriate transcription factor to reprogram them to get induced pluripotent stem cell that at this point can be again differentiated in the cell that you decided to study. Obviously, this approach has two different offer, two different opportunity. In this case, you get stem cells from patient to correct the defect and to reintroduce in the patient the cured cells. This is not our case. The system, the FOX2B system, is too much organized, complex. We cannot do this. We are not going to treat, to cure the disease by using, at least at the moment, to treat the disease by using stem cells, but we are doing this. We are modeling the disease. We try to have the CCHS in the plate because here, in this condition, we are very, very close to the disease and we can test the drug and so on, and we can understand pathogenetic mechanism. Basically, we have many patients who are donors of fibroblasts. We developed, as I told you before, stem cells from two different types of patients. We got fibroblasts. We reprogrammed cells. Everything was fine. All the different markers of induced stem cell, pluripotent stem cells were fine. And then at that point, we started to differentiate these cells from patients in sympathetic neurons. We have an intermediate passage because we have to get trunk neurons and then autonomic neurons. We did all of these different things, and finally we got the neural crest precursor, and finally sympathetic neurons, which are currently under study. One aspect is why we decided to study sympathetic neurons. These are sympathetic neurons. So is the efferent part of the autonomic nervous system. These neurons reach the different target organs and they regulate the activity of this organ. So many of the symptoms associated with the CCHS, beside the respiration, are probably related to malfunctioning of this neuron in the periphery. So for this reason, we have problem, temperature control, sweating, cardiac symptoms, and so on and so on. Probably if we can find something ameliorating the activity of FOX2B in these cells, probably we can respond to some needs of CCHS patient. We are not going to cure the disease. We are trying to ameliorating the symptoms. Next time, maybe in presence, I hope to tell you some new results in the field of CCHS, and let me show you my lab, which is not a big lab. It's a small lab. Fortunately, there are many women that are responsible for the research. Basically, I'm here to disturb. I'm kidding, obviously. Roberta Benfante, Simona Di Lascio, Silvia Cardani, who is in the Canadian lab right now, and Ana Lucia Quadros Gamboa, who in collaboration with Simona started this project on cells, on stem cells. And let me thank the different collaborators. basically located, with the exception of Silvia Pagliardini in Canada, all located in Italy. The association that funded our lab, and our gratitude is for ASIC, which is the Italian Association of the Family of CCHS patient, and the ASIC sustained our lab. Without their help, we couldn't do anything, and our gratitude to the family, to the patients, is very, very great. And thank you very much to everybody.